Okay, everyone. Thank you for coming. It's my pleasure to introduce Dr. April Wei, who is visiting us from Cornell. April is an assistant professor in computational biology at Cornell. And before that, she did a postdoc at UCLA and UC Berkeley. And before that, she completed a PhD at University of Michigan. And her lab does a lot of really cool things. And she's gonna tell us something about it today. So thank you.
Thank you, Amelia. So I'm going to start right away, and you're welcome to stop me with a question at any time. Oops. There it is. So today I'm going to talk about two projects developed in my lab related to graphs with genetic variation. Wait, I cannot. So to start, I'd like to introduce a coalescence tree to make sure everyone's on the same page. So this is a coalescence tree, and this axis is the time axis. From bottom up, it represents the time going from present to the past. And these leaf nodes represents samples or individuals that's related to each other through these common ancestor nodes. And the time of these common ancestor nodes represent how related to each other these leaf nodes are. And if they're related at this node, then it would be like an older common ancestor compared to if they're related at this node. And this is the root node, that is the most recent common ancestor. So for a species with recombination, we cannot use a single coalescence tree to represent the relatedness among individuals. Instead, we need a sequence of coalescence tree, and that is because recombination could happen in the past, which changes the relatedness among individuals. For example, so my cousin and I are related to each other through our shared grandparents, but in other parts of our genome, we could be as unrelated to each other as random people.
Yeah, I think it's very sensitive.
Yeah, I may need to let it sit in a way that's very stable. Yeah, so an arc could be seen. For the rest of the talk, you could think of an arc just as a sequence of coalescence trees that is changing along the genome. So the first ARG inference method that works with genomic data is called ARGweaver, and it's a very principled method using sequential Markovian coalescence process, and that models this recombination along the genome, and then also estimate the coalescence times. And that was published in 2014. And then we have seen a lot of ARG inference method over the past decade since ARGweaver, and most notably around 2019, the paper TS-Infer and Relate were published. And then later on, we also have more scalable ARG inference and also more accurate ARG inference. And before the ARG inference methods were available, demographic inference relies on using summary statistics from the genetic data. And this is a demographic model that represents the relationship among different populations: Chinese population, European population, and the African population. And one of the notable methods for demographic inference is called DADI, developed by Ryan Gutenkunst and co-authors, which utilize this kind of information that summarizes the set frequency in a two-dimensional space, comparing two population set frequency spectrum. And then they feed the demographic model based on this information to feed the observed set frequency spectrum, two-dimensional set frequency spectrum. But the whole information across many samples at many sites in the genome are reduced to this two-dimensional matrix. So the amount of information being utilized in this process is not a lot. If you imagine you have a single genome and you estimate set frequency spectrum, all you could get is the sites that have zero frequency, the frequency of 1, or the frequency of 2. So the amount of information we could extract for a single genome is really reduced to this three-dimensional. So even when we have a lot of samples, it's still a much reduced dimension. The amount of information we can utilize is very little. So since the ARG inference methods became more scalable and more accurate in the recent years, some new demographic inference methods have been developed, motivated by the massive information contained in ancestral recombination graph. So Gaia is a method that utilizes ancestral recombination graph to infer this individual's dispersal rate over time. And the assumption is that we can describe the individual's relatedness in the space as if they can disperse within a single generation, and then over time they would disperse in the space with common ancestor from the past. And GLAG is another method that utilizes ARGs to infer these admixture parameters, which include the population size of the ancestral populations and also the time of the admixture. And the assumption is that they could work with a population that's created through some source populations at a very at a recent time, and then with different proportion of ancestry coming from different source populations. And this is like a pulse of admixture event, where this is an event that happened at a single time point, and they want to estimate the time of the event and also the proportion from different populations. So, however, before our study, there hasn't been any ARG-based method that could estimate migration rates. And we think migration rates is an interesting parameter because we can think of different populations occupy different geographical space with migration connecting each other. And when we consider many subpopulations with migration connecting these subpopulations to each other is approximating a continuous space. Hence, it's approximating this dispersal of individuals across the geographical space. Whereas when we think about a lot of migration within a short period of time, it can also, on the other hand, approximate these pulses of admixture, creating a single population coming from different populations. So we're interested in this process, and we're interested in seeing whether ancestral recombination graph could be used in also estimating migration rates accurately. And we named our method mrpast that represents migration rate, population size across space and time. I'm going to talk a little bit more about this method later on. I think there's something weird with this sharing that I cannot move the slides very easily. It's very sensitive. So one of the motivation in using ARGs, and in particular that we want to use the coalescence time information in ARGs, is from this classical PSMC paper by Heng Li and Richard Durbin. So in this paper, the authors developed this method that utilizes a single genome, and then it's an HMM-based method with coalescence time information as the hidden states. So in some sense, they're estimating the coalescence times along the genome from a single individual's genome, and then they convert this coalescence time into this effective population size, because effective population size is really just the inverse of the coalescence rate. And they're able to estimate from this, each line here is representing their result from a single genome. And this x-axis is the years from present to past, and y-axis is reflecting the effective population size in the population that this individual's genome is sampled. So from each line, it's representing the history of the effective population size of a population. So from a single genome, they're able to resolve how the population size changes over time. And it's a lot of information they get from just having the coalescence time information from a single genome. So what we think would be interesting is if we have more genomes, and these genomes also come from different populations, we can also extract coalescence time information from an ancestral recombination graph. And instead of having a diploid genome where two samples are from the same population, that we have a coalescence time trajectory from that genome, we can have two samples that's from the same population, from this population or from another population, but we can also have two samples from across population. Say I have one sample from this population and one sample from this population. I can also ask in an ARG where does these two samples coalesce. And their coalescence time trajectory would be affected by not just the parameters affecting a single population, but also affected by whether a lineage has migrated in the past, that this lineage was originated from another population sometime in the past. So this change of color here represents that the lineage has migrated, that it was originally in one population colored in red, but then at one point it came from this other population colored in green. So with this information, if we can have pairwise coalescence times sampled from an ARG, and then if we can also predict based on a demographic model how the pairwise coalescence time should look like, we should be able to recover the parameters that determine this demographic process. So what we did here is really from an ARG, we can sample a single coalescent tree, and these samples we originally have from the data would be labeled with which population they come from. And then we would discretize the time into different slices and then count how many times a pair of lineages coming from one population would coalesce in which time interval. So for example, let's say these are our time slices we have. We observe this coalescence time T1. And this T1 is representing a coalescence between one lineage in population 1 and the second lineage in population 1. Therefore, we would add a count to this time slice in this data matrix that represent coalescing in time tau1, and then add 1 in the entry that's representing a lineage coalesce in pop1 and pop1. Similarly, we have this T2 that's highlighted here. So T2 represent a coalescence event that's coalescing this green lineage with 3 of the blue lineages, right? Therefore, it will add a count of 3, that is to this tau2 interval, that's representing a lineage in population 2 coalesce with 3 lineages in population 3, hence adding 3 coalescence counts in terms of pairwise coalescences. And similarly, it also represents this blue lineage coalescing with these other 3 lineages also in population 3 through this trajectory. Therefore, we also need to add 3, representing 3 additional counts that's a lineage in population 3 coalesced with another lineage in population 3. So therefore, starting with a single coalescent tree, we can keep adding the coalescence time information, summarizing it, and we can summarize it from many, many trees sampled in the ARG to get a data matrix that have information about how pairs of lineages coalesce. Similarly, we can build a theoretical model representing the rate of the changes in this stochastic process. So we have individuals from population 1 coalescing with each other. That's based on the coalescence rate in population 1. We can also have migration events that move this lineage backward in time to another lineage. And this change of lineage state comes from migration. So therefore, we can build up a stochastic process representing the same demographic process and then utilize this to compare with what's in the data. So we build this stochastic process and represent it in a rate matrix, and doing matrix exponentiation would then give us the transition probability into the coalescence states. And how fast this transition into the coalescence states would predict how many coalescence time we should observe in each time interval. Therefore, that's connected back to what we can observe in the data in the ancestral recombination graphs. So we collected these counts that's in each time interval, how many coalescence we observe between lineages sampled in the same population or in different populations. We also compute these probabilities based on a theoretical model that's predicting the density or the probability mass of observing a coalescence event within a time interval. And that way we can use a composite likelihood framework to calculate the likelihood of the observation given the parameters, and maximizing this likelihood would then give us the parameter estimates. So we have 3 modes of usage. First, we want to be able to accurately estimate the parameters using simulated ARGs. And then we would like to test using ARGs inferred from simulated data to understand whether there can be certain biases from inferring the ARG with a particular type of method. And I would use ms prime plus ts infer here to indicate the result shown is based on ARGs inferred from simulated data. And lastly, if we can accurately resolve the parameters based on this second approach, we would then test in real data. And I will use TS infer from 1,000 genome to indicate that is a result from in ARGs inferred from real data. So our data analysis pipeline is like this. Typically, we use about 30 to 50 unrelated individuals from each population. And we would also sample coalescence trees in low recombination rate regions, because these are the regions that there's more information for ARG inference methods to resolve the coalescence time. And we know that ARG inference is slightly more accurate in low recombination rate regions. And in real data, we also choose to use regions that's not as affected by background selection by using a threshold for B statistics. And then lastly, we would also bootstrap the coalescence trees to estimate uncertainty. So here's an example. In this example, we use the out of Africa model, and this is ARGs inferred from simulated, from ARGs inferred using inferred ARGs from simulated data, because we always get very good results from simulated ARGs directly. So all the results I'm going to show you are based on ARGs inferred from simulated genetic data or ARGs inferred using real data. So we infer ARGs based on simulated data under this 3 population out of Africa model. And this is the African population with a population size change at this time point. And then this population splits into 2 populations. This represents the out of Africa migration event. And then the European population has a change in effective population size. There's another split into the Chinese population. And then in recent times, there's also exponential growth in the European population and the Chinese population. And there are migrations connecting the 3 populations. So in total, there are 14 parameters. And this x-axis represent the ground truth parameter values, and y-axis represent the inference. And if they're sitting on a diagonal line, it means like the result is quite accurate. So these are the 4 migration rates, and these are the 2 growth rates. And these are 3 time epochs that we jointly estimated with the other parameters. And these are the effective population size parameters colored in blue. So overall, the accuracy is pretty high. We apply it to real data. The original model was proposed by the Daddy paper, which assumes symmetric migration. But when we apply to real data, we also wanted to test whether we can study asymmetric migration. And we see that oftentimes, if we estimate using a symmetric migration rate, the 2 resolved asymmetric migration rates, like this symmetric migration rate estimates is like in between the 2 asymmetric migration rates. But oftentimes, the migration is asymmetric, which in this figure is shown as this orange and the green boxes. And when they're like very different, it means like the migration is actually asymmetric rather than symmetric. There's different amount of gene flow from one population to another, then from another population to this population. And most interestingly, I think is this migration between the European population and the Han Chinese population. We see like migration direction is mostly forward in time from east to west rather than from west to east, and it's connected with some demographic process that is documented in the recent epoch. So next, we also test another model, or a model that is somewhat similar to what GLAC used, but GLAC could only do pulses of admixture, and instead we want to use a more realistic model, which include the growth of the population size and also the migration, but we also want to include this American admixture event. So we built a new model upon the 3 population out of Africa model with another split between East Asian and the Native American. And then in the recent time, we created a pulse of admixture represent 3 source population contributing to the admixed American population. And this model is somewhat special because we don't have actual data from the Native American. So in dealing with both the simulation data and in the real data, we want to use this model framework, but don't use any data from the Native American, which means that we wouldn't utilize the Native American and Native American coalescence or Native American against these other populations' coalescence events. So this model has more parameters. It has both migration and also admixture. It would also be interesting to see if these 2 processes would bias each other. But in overall, we see the results are pretty good. The crosses represent the parameters about the Native American population. So even though we didn't use any data from the Native Americans, we include the parameters from Native Americans. So we can also see that the Native American parameters could be resolved quite well as well. And this is like a plot representing the admixture proportions in the admixed individuals based on bootstrap results. And we can see overall the admixture proportion are quite accurately estimated, but there's a little bit of overestimation of the African ancestry. So in the real data, we estimated the admixture time for Colombians, Puerto Ricans, and Mexicans, and overall we see the admixture time is dated to about 20 generations ago. And we also estimated the time of the split between East Asians and Native Americans, and that's estimated to be around 7,500 generations ago. The effective population size estimated for Native American is very small overall, but with the caveat that we're using a single parameter to represent this, and there may have been a bottleneck, and there may have been a historical and the recent bottleneck, but overall, when we use a single parameter to estimate this, we see a relatively small effective population size. In terms of the admixture proportion, we see that the Mexican has the highest admixture proportion from Native American of about 30%, higher than the Colombians and the Puerto Ricans. So far, I've been talking about utilizing the coalescence time information from an ancestral recombination graph, but ancestral recombination graph is a lot more than just representing the coalescence time. For example, we can also have mutations represented on the branches of the trees. And then if we also represent mutations on the branches of the trees, ancestral recombination graph at the same time would also represent the genetic variation for all the samples this ARG represent. So it has been demonstrated by the TS Infer paper that this is like one way to compress the genetic data. But is it the best way to compress the genetic data? That's the question we're going to address in this next project. So we know that DNA sequences are this ATCG information. And for population genetics or statistical genetics, all we care about is this polymorphic sites along the DNA sequences. And then for a diploid genome, we can have a polymorphism of the ancestral or reference allele, a heterozygous individual, or a homozygous derived or homozygous minor allele. And this information could be also represented based on 0-1 information, this binary encoding, using 0 to represent one of the allele and 1 to represent the other allele. We can also use this count information that is most frequently used in statistical genetics to represent how many copies of the derived allele or non-reference allele that an individual genome has. So for this kind of information, it's the same kind of information an ancestral recombination graph could represent. However, in statistical genetics, what people usually do is to represent this data based on an n by m matrix, where n is the dimension of number of samples and m is the dimension of the number of mutations. And for present day biobank data, which contains a large number of human samples, n is on the order of 100,000 to 1 million, and m is on the order of a few hundred millions if we're talking about whole genome sequencing data. So for the UK biobank data, they have two releases. For the 200,000 samples UK biobank whole genome sequencing data, the size of the VCF, even after gzip compression, is approximately 1.5 terabytes in hard drive. And this is only in hard drive because when we analyze this VCF, this data has to be decompressed in the memory, then it will be even larger. And because of that, this data is only accessed through the cloud computing option, and it's very expensive to analyze this kind of data. So I mentioned that ARG could be a data structure, also represent the genotype, but ARG, on the other hand, is very expensive to build, and there are a lot of considerations and filtering in building the ARG as well. So the question we ask is, could we have a different format that can help us analyze this data, and especially if we can analyze this data and on a laptop, that could be nice. So for any binary matrix, we have two dimensions. And for the genotype matrix, we can think of one dimension as the samples and one dimension as the mutations or variants. And this is a binary matrix, and the genotype matrix is representing diploid. But if we think of it as we have two copies of genomes, one from dad, one from mom, in terms of phase genome, it can also be represented as a binary matrix. Any binary matrix can also be equivalent to a bipartite graph, where we use a bunch of nodes to represent one dimension. Say these leaf nodes represent the sample dimension, and the other dimension represent the variance dimension. Then we have connections between the variant dimension and the sample dimension to represent there's a one information. There's the information one. If there's no connection between a variant and a sample here, it would then represent 0 information. So connection in this graph represent the value in the matrix. And this holds true for any matrix, any binary matrix. So for a genotype matrix, we know that there's variant sharing. Some individuals, they're more related to each other, then they would share they would inherit the same mutation from a common ancestor. And that kind of variant sharing would then create this hierarchy that gives rise to this coalescent tree structure. It's a variant sharing structure as well. So what we think of is we should utilize this variant sharing structure and then build additional layers into this bipartite graph. And then at the same time, this additional layer would reduce the number of connections in the graph, therefore reduce the size of the data. And genetic data is naturally hierarchical because relatedness is hierarchical. And this relatedness gave rise to this additional sharing and connection. Then if S3 has a connection to V1, it could be connected through an intermediate node in between them. And by adding additional hierarchy, we can compress the data, but also represent the relatedness. This is a different graph from the ARG, and it's much simpler compared to the ARG. So what are we having in this graph? So we have nodes, and every node other than the leaf node represent the samples. And then we have these intermediate nodes and root nodes. And mutations are mapped to the nodes. Okay, and if a mutation is mapped to this node, is connected to these two leaf nodes, it means these two mutations are present in these two haplotypes. And singleton mutations are mapped directly to the leaf nodes representing the samples. So the rule of reading this graph is that if there is a connection between one node and a leaf node, that means all the mutations on this node would be carried by this leaf node haplotype. And mutation in this graph, quote unquote, is really like a variant. And it's encoded with the coordinate of the variant and the allele of the variant. So it doesn't have to be single nucleotide. It also doesn't necessarily to be biallelic. It could be multiallelic, and it could also be a missing information. So how do we read this graph? So from the mutation node, if we traverse down to the leaf node, we're able to read to reach all the samples having this particular variant, these two particular variants. So traversing down, we're reaching node 0, 2, and 3. That means node 0 and 2 and 3, they both have the one information for mutation 5 and mutation 10. And mutation 5 and mutation 10 represent these two original columns in the genotype matrix. We can also reach from the leaf node to the mutation nodes. So when we're reaching up, we're asking what are all the unique variants carried by this particular haplotype. And all the variants we reach as we reach this ancestral node are the variants carried by these unique haplotypes. That way we can recover both the variant to sample mapping, but also the sample to variant mapping. So at the beginning of last year, we published our first paper, also which include the concept of this graph that we call genotype representation graph, but also some initial results about this. And at that time, so our constructed graph is performing about the same as the other more complex compressed genomics methods. And for applying to UK biobank data, x-axis is representing different chromosomes of different lengths, and y-axis is the file size, and these orange dots are the original gzipped VCF. These blue color dots are the genotype representation graph size. We can see that we're much smaller than the VCF even after compression. So over the past year, we want to improve two things. One is the speed of constructing the graph. We know we could already speed up to like 1 million samples, but we want it to be even faster. Could it be so fast that people wouldn't need to be thinking about the cost of converting an original format to our format, such that it reduced the barrier of using this. And also we want to build infrastructure that could allow people to utilize this format more easily, because this format allows us to have the entire UK biobank data in RAM, and it can be analyzed in RAM. It can be used interactively in RAM as well. So this is a much improved version of the graph, which we're planning to preprint in 2 weeks. And it's also, in some sense, a very interesting algorithm that allows us to build the graph really, really fast. So we would start with samples that's really close to each other within a genomic window. Okay, so we will start with finding the samples that's really close to each other. And let's say they're represented as node I and node J. And sample haplotype is represented as a set of the variants that these two haplotypes carry. So that's represented as HI and HJ. That's the haplotype of them. So we build this graph by building a common ancestor node to pairs of samples that's really similar to each other. And these common ancestors, we then would propagate the union of the variants carried by sample I and J to this parent node. And because these two samples are really similar to each other, the vast majority of the variants would be shared, and then they would be propagated to this parent node. And for the remaining part of it, that's only in sample I, but not in sample J, we would propagate it to two additional nodes. And this node is only connected to I because these variants here are only carried by sample I. And the parent nodes plus the union of this plus the set difference would make sure that all the variants in sample I are still carried by node I. And similarly, we do the set difference. That's the variance only in haplotype J, but not in haplotype I. And then this set difference plus the set union would also give us this haplotype J back if we do the inheritance from this node and this node. And by doing this, we're able to build the graph, but at the same time know how the variants are mapped to the nodes in the graph. And at the end, we would also have an additional step that is taking, if a mutation is already represented by multiple nodes, we will need to combine it to represent a single mutation with a single node. But doing this is a single step in creating the graph, but also represent the mutation. And we get a graph that represents the same result as the original data. But that's the essence of the new algorithm. So how much does it improve compared to our previous algorithm, which rely on building trees and combining trees? So overall, we have this construction time that's improved more than 10 times. And construction cost is also improved. And the graph loading time has also been improved in this newer version. So we apply it to the more recently released 500,000 UK biobank whole genome sequencing data. And in this data set, there are 700 million variants we processed. So this x-axis is for chromosomes, and y-axis is the size of the graph for each chromosome. And we show that the new algorithm being applied to both the 500K and the 200K. And we see that for each chromosome of the 500K samples is taking like no more than like 10 or 11 gigabytes in size. And that's the same size the GRG will be when it's in RAM. So it can really be processed chromosome by chromosome with very little RAM usage. And how long does it take to build a GRG? If we use a single node with 70 cores, for each chromosome, it takes a few hours to convert from the gzipped VCF into a GRG. So and it only costs a few dollars for each chromosome. So we think this is fast, pretty fast, and is pretty low cost. So we compare it with Plink2 because Plink2 invented this new format that's called Pigeon that's becoming quite popular since it's also compatible with Plink. And we compare the cost of converting from VCF-GZ to Pigeon that people sometimes do when they process the UK biobank data or all of us data. And we compare it when we gave 2 threads to Plink2 versus 70 threads to Plink2 and versus our approach. And IGD is another format developed in my lab. And we can see that we're taking a lot less time. And for chromosome 22, we take about an hour and a half, whereas using Pigeon, it would take at least 3 times more than that. And if we use the IGD format instead of gzip VCF, is less than an hour. So I've been talking about constructing, but what are the other benefits of having this graph representation? And for one of them is that before, if we want to go through a whole matrix, we have to take n times m number of operations. And because the number of operations we need is to visit every entry in this matrix. Whereas if we have the same calculation done on a graph, all we need is to visit the connections and the edges. That is the size of the graph. And the size of the graph is much smaller than n times m. So any calculation in this graph space, if we can do it in the graph space, will be faster than doing it in the matrix space. And luckily, we can do things like the matrix represented by the graph multiply with a vector, and also the transpose of the matrix multiply with the vector by simply doing downward graph traversal or upward graph traversal. So this graph traversal, as we traverse the graph, we're also doing summations, and which gives us the mathematically equivalent result of multiplying this matrix with a vector. And that's what we can do. So, and the logic here is because as we do graph traversal, we're operating on this haploid genotype matrix, but putting things back from a haploid genotype matrix multiplication back into a diploid genotype matrix multiplication is another summation. And that's something very straightforward. And most of the things in statistical genetics rely on this simple operation that is a matrix multiplied with a vector. That is what people use in GWAS, that's what people use in PCA and many other analyses. And we want to allow StatGen people to use this new format as easily as they use other original formats. So we integrated with SciPy linear operator, provide operations for the genotype matrix and its transpose, and for the LD matrix or genetic relatedness matrix, either is standardized or is unstandardized. And we also build data filtering based on frequency, regions of the data, sample ID, something similar to what people use Plink for, that is to QC, do quality control of the data for their downstream analysis. And is now both a command line tool and also a Python library. So I'm going to show you how much it can speed up things. So comparing with, so this is for doing GWAS with covariates, comparing with Plink 2 using pgen, when we use one thread, we're comparing this blue bar with this green bar. And x-axis is the number of samples from 100,000 to 1 million. And y-axis is in log scale. So you can see that when sample size is large, we're more than 100 times faster than using pgen. And if we give pgen 25 threads, we're still faster than that using a single thread. And in terms of memory usage, because we have to load the graph entirely into the RAM, whereas pgen could stream in the data one by one, we see that we're using like 2 to 3 times larger RAM than pgen. But overall, because the graph size we have is so small, we're still using on the order of 10 gigs of RAM for this calculation. And this is the p-value we have comparing to Plink. It should really be identical. So this minor difference around the very large p-value that people may not care about that much could be due to some numerical instability. This is a benchmark against the software called TS Trait, that is an ancestral recombination graph-based phenotype simulator. And so for doing the same thing, compared to ancestral recombination graph, when the sample size is large, we're also orders of magnitude faster because this is y-axis is log scale as well. So another thing people usually do when they have some data is they may want to do a principal component analysis on the data. And we implemented 2 types of PCA. One of them is identical to the Plink PCA, and another is probabilistic PCA that's supposed to be a little bit faster. And we benchmark with both Plink PCA and Flash PCA too. So let's just look at this blue color bar. against the green and the purple. We can see that generally speaking, the GRG PCA is at least 2 orders of magnitude faster than Plink, and Flash PCA2 is getting too slow, so we didn't benchmark it for 1 million samples. We apply it to bank data, only filtering out the allele counts to be less than 20 in total in the 500K samples, applying it to the whole genome sequencing data. And it takes a few hours to get 10 or 20 PC for the whole UK biobank. So how easy it is to implement. The PCA method I just showed you was implemented in 4 lines of code after we built the infrastructure. It is now the fastest PCA method. So all we need is to import the GRG, 2GRG library, and then import SciPy. And then we have 4 lines of code leveraging the SciPy method to compute eigen decomposition. And that gives us the identical result to Plink, but is orders of magnitude faster. So, so I hope this will be something that enable many methods in StatGen to be really very easily implemented with what's already there in the Python ecospace. So in conclusion, I talked about 2 types of graph representation of the genetic variation, ancestral recombination graph and genotype representation graph. And we talked about ARGs can be quite useful for demographic inference because it really has a lot of information about the relatedness among individuals, the time information. And we also talked about genotype representation graph is somewhat related to ARG in the sense that it utilized the information of relatedness to only represent the variant sharing information. And we talked about how it can be constructed and how it enables faster linear algebra calculations. So with that, I'd like to thank the people who did the work. So Drew is the lead author for both work actually, and the demographic inference work was mostly done by him, and with the help of an undergrad student, Anthony, and a collaborator, Leo. And Drew and Ziqing and Chris and Aditya, they all worked on the GRG related work. And all the softwares could be installed via Bioconda very easily. Yeah, and thank you. And I'd like to take any questions you have.
Yeah, it was a really interesting talk. I worked on malaria lab, and an issue that we often run into is with like our data set, genomic data sets and storage, we have like with BCFs, and the common issue is like we'll have like a large multi-sample BCF, we want to do like some new analysis with an additional sample set with like integrated and build on. If you're using this graph-based storage, how easy is that, like iterative, of reading a new sample to an existing graph?
Yeah, that's a good question. So I think it will be much easier than doing VCF. And for one, that is the I/O process is much simpler, and loading time is much shorter. We haven't built in ways to add in new samples, but it wouldn't be extremely hard to do. We did already build in filtering of old samples and filtering of old variants, but I think it will be, yeah, it will be interesting to also add that. Yeah, thank you. Okay.
Thank you for this talk. With regarding mrpast, how does the transition model like take into account that you're kind of, I guess for lack of better words, like skipping over loci if you're just sampling from low background selection regions and low recombination regions?
Yeah, so good question. So what we assume is a composite likelihood. That means we're assuming we're sampling independent trees. So yeah, so that's an assumption we made. Yeah, in a sense that we're not considering the recombination between loci, but that would be an interesting extension for the future.
Sharon, in your GRG patient, have you ventured into how we can like know features to graphs, such as like functional related data? They're like, I guess like data that you have, you could attach them to your file. Kind of affect the implementation.
Yeah, that's a good question. So for now, any annotation in addition to the allele information is considered that could be loaded from a separate file. Because in some sense, that's not the information we can compress. We can put it aside in a separate file, or it can be loaded in to be co-analyzed with the GRG. For example, we can imagine like a phenotype file, right? Would be like for each individual. It's not necessarily something we want to store in the GRG because it's not going to save a lot. And those are the things people would operate very differently depending on what they work on, whether they care about the annotation for the variants or the annotation for the individual. So that should be like better set aside. That's very similar to how Plink handles things, and yeah.
One thing's given like it sounds really cool, those graph representation. What are the things that you would not be able to do?
Yeah, it's a great question. So so what we already have are GWAS and PCA, ridge regression, fine mapping, and what we think we can do as well for nonlinear stuff is logistic regression. That we have to think of how to map the nonlinear operations into linear operations, because right now with the graph, the best thing we can do are linear. There are certain nonlinear operations that people already figure out how to map it to linear operations that we can borrow directly. But there are also things like, there are people who do like this deep neural network on genotype matrix with these filters that's scanning through the genome, right? That is very hard to find something that's exactly equivalent. But there may be some other machine learning methods that we can do. That wouldn't be the same. Like we wouldn't. So, in some sense, it's the same data, but there are things that we can do faster than the conventional approach. But there are also things that we wouldn't be able to do faster than the conventional approach. And we're very interested in, figuring out how to do nonlinear stuff on this graph.
I'll start with one of the challenges of using multilinear mixed models is having to pass the GRM for the one. So we think, maybe GRG there. It should be similar to what's going on.
Yeah, exactly. So what we have are these like, yeah. Yeah, so what we start with is this linear operation with respect to genotype matrix. But if we do 2 times of this, that gives us this linear operation with respect to LD or GRM. And we can also do standardization, because standardization is subtracting and dividing element-wise, so that is also linear. So what we build in is, if it's an operation, say, like something that only use GRM in the sense that you need a linear operation against the GRM, that we can do. But to compute a GRM directly would still be hard. Like something like RAML would need would still be very costly because you need the GRM directly. Some of the RAMO method need the GRM directly, but some other RAMO method or approximate RAMO method would only need a linear operator against the GRM, or then it's fine.
I'm curious about whether there are operations from the perspective, I guess going bottom up, where you could learn something interesting about variants, right? So if you have some variants that have many, many children, that don't, like, would, you know, think about, I guess the challenge of this linear operation, but I'm wondering if there's something about the structure of the GRG itself that can help us understand elements under strong relative selection.
Yeah, that's a great question. So we can define a GRG from an ARG. And in fact, if we already have an ARG, we can convert it to a GRG, like taking seconds at most. So in some sense, there's a better GRG. That's not the GRG we're building right now, because we're just building a graph that represents the data very fast. We don't do all the checks about whether it's population genetics relevant or not. But I'd be really interested in doing that to see whether we can get a GRG that this hierarchy actually means something. Like right now, it doesn't. It doesn't always mean something. Yeah.
Could there, I guess following up on that, could there be like graph features, you know, like people copy things like something about like the nodes or how well connected they are, or something about the features of the actual graph? It could actually be meaningful in terms of like representing genetic variation.
Yeah, like, sorry to add on, but aren't the root nodes at least like interpretable in some way, or the degree of the nodes?
Yeah. So we tested like whether these like nodes with more layers are older, and they are. And this nodes that's like in the shallow part of the graph, they're also younger. But it's just like right now they're not super useful. Yeah, it's in the sense that it's cool.
Any more questions? Thank you so much.